Bismillahirrahmanirrahim. Assalamu alaikum everyone. This is Masab Umair. I'm working as a senior scientist and in charge of virology department at NIH. So I will present our findings on a study which was based on the genomic characterization of respiratory viruses, mainly influenza virus, SARS-CoV-2 and RSV. So respiratory illnesses, they are a major public health issue in Pakistan, mainly affecting children and elderly above 60 years of age. So some of the common respiratory viruses are the influenza virus A and B, RSV, SARS-CoV-2, and human metapneumovirus. Although respiratory viruses, they are being surveyed in Pakistan through different programs. So we have a National Influenza Center that is doing the surveillance for these viruses, and then also there are some studies published. But there is not such a study or a data available on the integration of these three viruses and its prevalence in the population. Also, the previous studies and surveillance is mainly done on PCR based and on sequencing of partial segments of these viruses. So with this limitation, we investigated the prevalence of these three viruses, and we also explored the genomic diversity based on whole genome sequencing. So during 2023 and in the first month of this year, we enrolled 320 patients who had respiratory symptoms. So respiratory samples such as oropharyngeal, nasopharyngeal swabs, they were taken, and viral nucleic acid was extracted using commercially available kits. Initially, the detection of the three viruses was done using a triplex PCR kit, and the positive samples that had cycle threshold value of less than 30, they were further subjected to whole genome sequencing. So we used a commercially available Illumina RVOP kit, which targets these three viruses as well as some of the other viruses. So after library preparation, the library quality and size was checked, and the library was loaded on to the Illumina MySeq. And the FASTQ generated from Illumina MySeq was then further analyzed using the bioinformatic pipelines for downstream analysis. And we also performed the phylogenetic and mutation analysis using different softwares. So the results showed that a high percentage, 57% of the cases among our patients were positive for either one of the three viruses. So majority of the infections were caused by influenza virus, followed by SARS-CoV-2 up to 30%, and similarly we found 21 cases of RSV. This month-wise distribution shows that influenza was mainly found during the winter season, so peak cases were reported in November and December, whereas SARS-CoV-2 cases were evenly distributed across the year, and RSV was also found mainly during the winter season. So among the 183 positive cases, so we subjected 100 samples for whole genome sequencing, and the criteria was basically the CT value. So all the samples with CT value of less than 30, they were subjected to whole genome sequencing. So based on the whole genome sequencing, we managed to retrieve the whole genomes of 85 samples. This included 41 whole genome sequences of influenza virus, 39 whole genomes of SARS-CoV-2, and 5 respiratory syncytial virus sequences. And the whole genome sequencing results, they showed that among SARS-CoV-2, there were several different subvariants of Omicron that were detected in our study. And the major or the common Omicron subvariant was GW.5. Similarly, we also found XBB and its subvariants. So this GW.5 is one of the subvariants of Omicron that has those flip mutations, which basically enhance the virus ability to bind to cells and infect people. So we found these flip mutation in GW5 in Pakistan strains. For influenza, we only found influenza type A virus, and among the subtypes, so the predominance was the H3N2, and we also found H1N1 subtype. Among the H3N2, the major clade that was reported was the 3C2A1B 2A2A3A.1. And this clade was reported in 2022 in UAE, and then it later displaced other clades of H3N2 globally. Similarly, for H1N1, there was one clade which was reported, and again, this clade has been found in 71% cases of H1N1 reported globally since 2023. For RSV, so there are two, there are two subtypes of RSV, RSV A and B. So we only found RSV B in our study, and the genotype that was reported was GB505A. So looking at the phylogenetic analysis for influenza, the outer glycoproteins, hemagglutinin and neuraminidase, they are basically used for phylogenetic analysis. So based on the HA and NA sequence of H3N2 strains, we found that our strains, they were mainly, related to strains from USA, UK, Russia, reported in 2023. And here Pakistani strains, they are the current study strains, they are shown in light blue and dark blue triangles. Similarly, for H1N1 phylogenetics, you can see that as our strains shown in dark blue triangles, they showed close homology with strains that were circulating globally at that time. Looking at the mutation profiles of H1 and H3 and 2. So in H3 and 2, especially the most dominant clade that I mentioned previously, we found some unique mutations in the HA segment. And these mutations, they have impact on viral resistance to antivirals and as well as on viral fitness. For H1N1, again, there were some unique mutations that are highlighted in red here. So these mutations, they also are involved in, viral fitness and immune escape. Our SARS-CoV-2 phylogenetic analysis, it showed different Omicron subvariants that were closely related to strains that were circulating at that time, and we also found some introduction of the viruses into Pakistan from different countries. For RSV-B sequences, as I mentioned, so we found the GB505A genotype, but it was further segregated into different clusters. So two of the clusters, BD411 and BDE.1, which is the one in which our strains, they clustered, and these strains, they showed homology with some of the strains that were reported globally. We also found some unique mutations in our RSV segment G and F, highlighted again in the red. And these mutations, they also have implications on the resistance to different antivirals and on viral fitness. So in conclusion, our study showed high prevalence of respiratory pathogens. The three viruses that we basically look for, influenza was the predominant strain, then virus, and then SARS-CoV-2 and RSV, which highlights that these viruses, they continue to pose a significant public health challenge for Pakistan. Looking into the genomic inside, we found different diverse clades and subtypes of influenza virus, similarly for SARS-CoV-2 and RSV-B, which is again crucial for understanding virus evolution and its impact on vaccine efficacy. For mutation analysis, we found significant mutations, especially in HA gene of influenza virus and F and G protein of RSV, and similarly in the spike protein of SARS-CoV-2. So this again highlights the potential impact of these mutations on the antiviral treatments and on the vaccine efficacy and immune evasion. So it's again important to continue doing the surveillance of these viruses. And lastly, I think our study highlights or calls for enhanced surveillance, especially using integrated genomic surveillance of these three viral pathogens, in addition to other viral pathogens. So in order to effectively monitor these respiratory viruses and to improve public health response to emerging threats in Pakistan. Here I mentioned my email address. If there are any questions, I will be happy to answer them. Thank you.